Okay, so I guess to introduce, let's a couple of overall things. So this isn't going to be about teaching you material per se. There's going to be some content on here, but mainly going to be to add to your analytical toolkit. So there's different types of problems that are common, and recognizing them and knowing how to solve them within all the topics and subjects that there are on the MCAT. That's the goal for today. And as far as like content, I always say start out with the AAMC website. It's got the super exhaustive list of topics, the breakdown of topics as far as like percentages and the bigger categories, and then other logistical information about how this section works. And so how I usually do presentations, for those of you who are new, is I usually like to, I usually pay attention to the chat. I usually like to take questions as they come. So if you have a question, go ahead and put it in the chat. And I'll get to it as soon as I reach like the end of a sentence or a logical stopping point. So feel free to interrupt me with questions. Nobody's here to judge on what questions you ask. Yeah, feel free to ask questions at any time, and I will do my best to answer them. So with that, let's get started. So in the biology and biochemistry section, you'll often find like there's a limited number of possibilities for like what kind of experiments they can hit you with as far as analyzing a paper, for example. So one common one is a rescue, which is a form of phenotype replication. So essentially, the goal is to explore the cause or mechanism behind disease and how it might change a person's phenotype. So maybe someone's missing an enzyme, so they can't process a particular nutrient, or they're missing a key molecular factor. Essentially, something that leads to a visible effect at some level physiologically. So, for example, let's look at hemophilia. Hemophilia is a clotting disorder. So it's a defect in the gene for a protein called factor VIII, and that's a protein involved in clotting, so it doesn't work. So if we didn't know the cause of hemophilia, but we suspected that it might be factor VIII or problem with factor VIII, we might run an experiment. So our experiment would be we'd have normal blood, a hemophiliac's blood, and then a hemophiliac's blood with purified factor VIII obtained from somewhere else added. So here's our experiment. Got a normal mouse, and so if we were to have a small laceration of the blood vessels, we'd see normal clotting. A hemophiliac mouse would show poor clotting, which would make sense. And if we had a hemophiliac mouse and then injected factor VIII into the bloodstream, and we saw normal clotting, that would mean that factor VIII is that key feature that is missing in a hemophiliac individual. So this is an example of a rescue experiment where injecting that factor VIII protein is actually rescuing the hemophiliac mouse and allowing it to clot its blood normally. The other type of phenotype in replication experiment is a disease induction. So instead of trying to change something and fix the disease, you're actually changing something and causing the disease. So hemophilia, like before, we talked about how factor VIII is the key. So let's say we're trying to figure that out. We might say, okay, if we break or knock out the factor VIII gene, will that induce hemophilia? And so we might run that experiment. So a normal mouse, normal clotting, hemophiliac mouse, poor clotting. A normal mouse where the only thing we change is knocking out or messing with the factor VIII gene, if that shows poor clotting, which it would, that would mean that the factor VIII protein, which is the gene product of the factor VIII gene, is important, and that missing that or having a defect in that can cause hemophilia. Another thing that can be done kind of along these lines are a domain deletion. So proteins, usually proteins, even if they're made of one single piece of polypeptide, one chain, they can have different regions that are important for activity. And these regions are called domains. And domains are usually separated by flexible regions that don't have a particular direct effect on the protein function. So factor VIII, the protein that we've been talking about, has 5 domains: A1, A2, A3, C1, and C2. Now let's say we knew that factor VIII was important for clotting and the defect in it would cause hemophilia, but we didn't know which part of it. What we could do is we could take a normal mouse we would take a normal mouse and knock out the factor VIII gene. Now, normally that would lead to poor clotting, but let's say we added back the factor VIII protein or the factor VIII gene with one of the domains missing. So now suddenly that A3 domain isn't there anymore. And then we can examine, like, okay, does this domain deleted version of the gene, version of the protein, still rescue that hemophiliac phenotype? If it does, that means that maybe A3 wasn't so important in directly involved in clotting. Now, if this modified version of factor VIII is unable to rescue the hemophiliac mouse, then that means that A3 is probably important for clotting. Does that make sense? Say yes or no in the chat. Okay, excellent. All right, we'll keep going. Okay, combination studies. So this is where you're going to have one of those graphs with a table underneath it that has a lot of minuses and plus. So you have a bunch of factors and you're combining them in different permutations. So this can be kind of intimidating, especially if there's a lot of molecules and proteins that they're throwing at you. So, but the way to do it is you first analyze it like any other graph. So axes, okay, it's a bar graph, so there's individual discrete treatments. The y-axis is mRNA levels of a particular gene as a percent of the control. And notice that that first column is control, so it's at 100%. That makes sense. And then now the question is, what might be the purpose of each treatment? So that can tell us what this graph what conclusions might be important to draw from this graph. So for example, the first bar is the control because it's got minus, 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 nothing added. Bar 2 versus bar 1 shows us what happens when CTS is added. And it doesn't really matter for us right now what CTS or LNAME or TNF alpha is. But the point is that there's just 3 different things that are added. So bar 2 versus bar 1 is CTS versus nothing. And the only thing that changes there is the CTS. Bar 3 versus bar 1 shows what happened for TNF alpha versus nothing. And then bars 3 through 5 compare TNF alpha alone, like in bar 3, with TNF alpha plus CTS in bar 4, and TNF alpha plus LNAME in bar 5. And so maybe this, even without seeing the passage, we might say, okay, this is looking at some sort of synergistic effect, or they're looking at interactive effects between TNF alpha and then each of these other 2 things. Maybe they're in opposition. Maybe they're, we see first of all that TNF alpha by itself is causing a huge drop in mRNA. But if we add CTS or LNAME, we see a partial restoration of those mRNA levels. So maybe CTS and LNAME is working against TNF alpha. So we can get a lot of information if we systematically go through this type of figure. Okay, there's another experimental strategy called mutagenesis, sometimes called site-directed mutagenesis. So I mean, the mutation notation for a protein usually goes like this. It's a letter, a number, and then another letter. And so how to read that is, so you've got your protein that has a whole bunch of amino acids in it. And so if you have the E652K version of that protein, all that means is that at position 652, a glutamic acid, which is abbreviated as E, has been replaced with a lysine, which is abbreviated as K. And so when you're asked to judge what a mutation might be doing, you need to look at you're comparing the amino acids. So if the amino acids have similar properties, maybe they can perform similar catalytic or electrostatic functions. So if a glutamic acid is usually negatively charged, maybe it is stabilizing a positive charge in a substrate. So if you lose that and don't have a negative charge, that's going to mess with the enzyme's activity. But if you replace glutamic acid with maybe aspartic acid, which is also negatively charged, maybe that doesn't result in such a huge problem. That being said, switching out one amino acid for the other, even if they have similar like charge properties or polarity, might still be an issue if the binding pocket has to be a particular shape. Now, for amino acids with different properties, like glutamic acid and lysine, which are negatively and positively charged, if the mutation is in an important place, that can be detrimental to the enzyme's activity, because now you're losing a potential interaction with the substrate or catalytic activity. So let's look at a few examples. So if we had a mutation at E522D, that means at the 522nd position, we have a glutamic acid being converted to an aspartic acid. Those are pretty similar, and they're roughly the same size, so maybe that isn't going to be so bad. G332P, okay, that's switching a glycine at 332 to proline. Glycine and proline are very different. Glycine is much smaller, and proline also, because it has it's the only amino acid with a weird ring shape that includes the backbone, it might introduce structural limitations that can be damaging to the enzyme's activity. Okay, Y191F. Y is tyrosine, F is phenylalanine. They're similarly sized, but tyrosine has a key OH group that's sometimes modified by kinases. And so if you lose that site, that might be a problem for the protein function. And the last one, I231L, that's isoleucine to leucine at position 231. They have similar properties because they're both nonpolar. They both have the same number of carbons, a slightly different shape because they have a little bit different connectivity. Maybe they can be substituted without too much of a problem. So you can use this type of reasoning to analyze when you see a protein versus a mutant version of that protein, what you might expect in terms of activity and what has changed for that protein. Okay, vehicle control. So you'll often see in experiments when they're delivering a drug or a virus, or they're delivering a drug or a gene into a cell or an organism, they'll use a particular vehicle or vector. So that can be like a liposome or a plasmid or a virus. And so when they're looking for an effect of that drug or that gene, they want to make sure that the effect is due to the actual cargo, that being the gene or the drug, not the vector, the liposome, plasmid, or virus. So they always include a trial with an empty vector control. So just the vector with nothing in it, and ideally, your vector should look exactly like doing nothing. And if your vector does have an effect, you want to make sure that's not going to interfere with or give you a false positive for an effect that you want to see due to the cargo. Okay, before we move to principles, any questions? If you have no questions, just say no questions. Okay, only 3 people said anything, but they said no questions, so I'm going to continue. If you want to go back, feel free to put a message in the chat, and we can go back and take a look at something if you want it clarified. Okay, principles. So this is actually a really useful principle that I found when they're talking about like multiple genes or proteins working at the same time, different regulation pathways or like stuff like that. So, for example, there was a passage that I saw that was describing like a model of cancer, and it was talking about all these proteins that are involved, and so they were asking questions about what you'd expect if you inhibited or activated the proteins in a certain way, and so usually a lot of information to parse, especially if you're not familiar with the details of cancer biology, which most people aren't. And so I think one of the things that I found helpful is you essentially split these proteins or genes or effects into teams. So in the cancer example, you're going to have some proteins or some regulatory effects on team cancer and some on team not cancer. So like if they give you these 6 proteins, P53, RB, and P21 are on team not cancer. c-Myc, Circ, and AKT are all on team cancer. Now, with these assignments to these groups, we can then answer questions and draw conclusions. So, for example, which of the following would reduce uncontrolled cell growth? So we can first understand that just means not cancer. So you're looking for something that's an effect that is on team not cancer. So inhibiting c-Myc, that's against something on team cancer, so that's not cancer. Activating something on team cancer, that's cancer, so you don't want that. Inhibiting something on team not cancer, that's going to be pro-cancer, so you don't want that either. And then activating something else on team cancer, also not what we want. So that gives us the first answer. Yeah, so yes and no. So mostly you'll be able to determine based on the passage. So usually in the passage, you're going to be given all these proteins because you shouldn't be expected to know these types of a lot of these proteins. And you'll be able to tell and put that together based on what the passage is talking about. That being said, some of the proteins on this slide are good to know regardless, because then you can have a little bit of an edge. Yeah, like if you know what P53 is going in, and they try to explain what it is in the passage, and if you already know, that'll save you a little bit of time. So I would say when you're practicing and you see like common proteins, and it's kind of hard to tell which are common and which aren't if you haven't seen them before. I would say good rule of thumb is for specific proteins that are not like part of a biochemistry or cell biology course that are pretty niche, you don't have to memorize those. Some are just useful to know, but like even if you don't know what any of these proteins are, you're not going to be in huge trouble. But I would say, at least on this list, these 6 are nice to know. Just for clarification, the ones on the left are oncogenes, the ones on the right are tumor suppressor genes. Yep, very good. Okay, another principle is gene knockout and knock-in. So if you recall the rescue and disease induction experiment, we can either knock out a gene or knock in a gene. Knocking out a gene means you eliminate that gene from the genome, or at least mess it up enough that it's not transcribed properly. And so if you have a mouse or a cell line that has a gene knocked out, it'll often be shown as delta gene name. So like a delta P53 model would have an absence of P53, and so it'd have a greater risk of cancer. Now, a knock-in is a gene that wasn't in the genome before, but it replaces something that was there. So if you're trying to introduce a gene, you would knock it in. Okay, so those of you who were at my previous session for MCAT overall will remember this. Necessary versus sufficient, a huge deal in understanding and drawing conclusions about biological processes. So necessary means if you take B away, I'm sorry, if you take A away, B does not occur. A is necessary for B to occur. That means that A is the only cause of B. Now, if A is sufficient for B to occur, that means that if you add A and nothing else, B does occur. So A will always cause B. So let's look at a few examples. So let's look at something that's necessary and sufficient. So a particular gene mutation is the only cause of and always causes Tay-Sachs disease. Now, no one without the mutation gets the disease, which means that it's necessary. And if you have the mutation, you will always get the disease. That's sufficiency. So this is necessary and sufficient. Necessary but not sufficient. So being infected with HIV is the only cause of AIDS. No uninfected person ever gets AIDS, so it's necessary. But you can get infected, but you might not get AIDS. So being an HIV infection is necessary for AIDS, but not sufficient for AIDS. Okay, sufficient but not necessary. So decapitation is not the only cause of death, but it pretty much always guarantees death. So not all deaths are caused by decapitation. So decapitation is not necessary for death. Death can happen in a lot of other ways. But a person who is decapitated is probably dead, meaning that decapitation is sufficient to cause death. Now, something that is neither necessary nor sufficient, a number being odd is neither necessary nor sufficient for the number being a multiple of 5. So if a number isn't odd, it can still be a multiple of 5, so it's not necessary. And a number can be odd but not be a multiple of 5, so it's not sufficient either. Okay, let's talk about feedback. So there's two types of feedback, positive feedback and negative feedback. In positive feedback, the product of a pathway causes the pathway to become more active. For example, oxytocin, the more you produce, the more the pathway is activated, the more you produce. Negative feedback, the product of the pathway causes the pathway to be less active. So the more ATP builds up, the less glycolysis occurs because there's negative regulatory mechanisms that slow down glycolysis when there's an excess of ATP, which is the product. So positive feedback, if you got this reaction pathway, A to B to C to D, D will go back and amplify the activity of the reaction A to B, which will in turn result in more D. Negative feedback, where the same reaction, A to B to C to D, D will actually go back and inhibit the A to B reaction, thereby producing less B, less C, and eventually less D. And then if D is being consumed by something else, D will eventually be depleted, that inhibition will stop, and the pathway will start back up again, producing more D. Is the amplification linear, exponential, or something else? I think it depends on the reaction. I don't think you need to worry about that. I think just understanding how this works. So if somebody said, if you were asked about a negative feedback pathway, you would realize that an excess of D would actually lead to less B and C, because now you're blocking that first reaction. Therefore, less B and C are produced. Yes, okay, that's a great example. So yeah, ATP is a allosteric inhibitor of hexokinase. So if you have a ton of ATP, you don't have to do glycolysis very much because you have enough ATP. So you don't have to devote resources toward that pathway. But if you're low on ATP, hexokinase will be active, and therefore you will do glycolysis and produce ATP. So thanks for that example. Okay, manipulating gene expression or protein production. So these are experimental tools that you'll often run into. So if you want to stop the expression of a gene product at the DNA level, you would do something like a gene knockout, where you could do something like CRISPR, where you'd essentially break the gene so the gene isn't transcribed, the RNA doesn't do anything, or it's not produced, but there's no protein. Now, another way of doing it, if you wanted to silence a gene, like at the RNA level, which is a lot easier experimentally than knocking out a gene, you could use siRNA or miRNA, which are basically oligonucleotides. So you'll have these it stands for small interfering RNA or microRNA. And what they do is they bind to the RNA if they're complementary to it, and they will essentially tag it for destruction, preventing it from being translated. So the DNA is still being transcribed, but the RNA is destroyed by siRNA or miRNA binding before it can be translated. And then if you wanted to mess with a protein after it's produced, you'd use something like a specific inhibitor. So you'd be able to inactivate the protein once it's already produced. Okay, let's talk about structures you should know. And this is by no means an exhaustive list, but it's the ones that I thought of after thinking for a while. So DNA versus RNA. So you should know how to identify deoxyribose versus ribose. So you should be able to tell whether a given nucleotide is DNA or RNA based on whether it has that missing oxygen or not. You should be able to identify the bases A, C, T, G, and U on site. And you should also be able to know where the 5' and 3' carbons are, so that you can be able to identify where that chain is going to be forming when DNA is polymerized. Okay, amino acids. Amino acids are one of the most important things for this section. You need to know all the amino acids, all the 20 amino acids. The side chains, you should be able to draw them from memory very quickly. And you should also know the properties. So which ones are acids, which ones are bases, which ones are hydrophilic, hydrophobic, and with a particular emphasis on potential binding interactions. So let me give you an example. So DNA is negatively charged because of all those phosphate groups. So you might expect the active site on a DNA polymerase, which binds to DNA, to have a lot of positively charged side chains like arginine, like lysine. And so therefore, if they ask you, like, okay, DNA polymerase active site, which amino acid would you expect? It'd be, you'd expect to pick one of the basic ones because it's positively charged at physiological pH. Positively charged will help bind to a negatively charged DNA backbone. Okay, sugars. Common monosaccharides are good to know, both in terms of Fisher projections and in terms of what they look like on I don't know what those are called, the other 3D structure, the cyclic structure. That can be really useful, and I personally think it's essential. There's some really good mnemonics for knowing how to identify each one, as far as like which ones. So they have a particular uniqueness as far as which oxygens are pointed up versus down. And so there's mnemonics. Reddit is a pretty good resource for this. Like Manos is like, it's like a gun. So like carbons 2 and 3 are going to be, oxygens 2 and 3 are going to be pointed up, 4 and 5 pointed down, stuff like that. I'd also recommend knowing what the alpha and the beta versions of a cyclic monosaccharide are, as well as where the anomeric carbon is, and the L versus the D sugars, and you should know the fact that we use D sugars and L amino acids. And now I'm actually gonna go on a slight tangent here because I thought of this. So we use L amino acids. All of the amino acids we use have S stereochemistry, except cysteine, which is R stereochemistry. But all of them are L. So that can be a really useful tidbit to know. I'll say that one more time. We use all L amino acids, which are all S except cysteine, which is R. And we use D sugars. Okay, there's a question in the chat. Should we know the pKa for the amino acids? I would say you should be able to estimate with some degree of accuracy. Like you should, like, yeah, like kind of like you said, the acidic ones, pKa is around 3 or 4. Basic ones, it can go from 8 to 10. I don't think you have to know specifically what they are. You should just be able to ballpark it. Yep. Okay. Lipids, you should know phospholipids and triacylglycerols, what the structure looks like, and how, especially for triacylglycerols, you should know how to what reagent might be used and how it works to separate it into these 3 fatty acids plus the glycerol. That's also useful to know. Okay, loss of function versus gain of function. So a loss of function, usually in the form of a mutation. So you'll see like loss of function mutations or gain of function mutations. So a loss of function mutation is when something stops working as intended. So a tumor suppressor gene, if you have a loss of function, you might have an increased risk of cancer. So like, let's say for example, classic gene, like P53. Let's say that one of your parents, you inherit from one of your parents a defective P53 gene. You have one copy that still works. All amino acids are all amino acids. Yes, so all amino acids are S except for cysteine, which is R. I'm 99% sure that that's what it is. Feel free to fact check me on that, but I'm pretty sure that's what it is. Okay, so let's say that one of your parents, you inherit a defective P53 gene. Your other one is fine. But let's say that that other one acquires a loss of function mutation. Now you have no P53 in those cells, and now you have a significant risk of cancer. Now, a gain of function is when something starts working in a certain way, possibly not as intended. So for example, an oncogene, if you have a gain of function, that might result in an increased risk of cancer. So like one of the ones we were talking about before, like SERC or CMYC or AKT, if you have a gain of function that causes them to be hyperactive, that can mess with, that can increase your cancer risk and mess with cell growth. Yeah, so let's look at these examples. Loss of function. So let's say someone has 2 good copies that are producing gene product. One of them is mutated, so it has loss of function. So now you're only producing half of it. And if you were to lose both of them, which is pretty unlikely, but if you already have one that's not working, and then you lose one, you will now have a totally inactive gene. So you will have a loss of function could be more severe that way. Now, gain of function. So gain of functions tend to be dominant, whereas loss of function tend to be recessive. So usually takes two alleles that have experienced a loss of function for it to matter. Gain of function, usually 1 is good enough to cause problems. So mutated gene would be dominant. It would produce a defective or hyperactive gene product. And that could, it's already bad enough with 1. If it's 2, it's even worse. So that's how gain of function works. Okay, let's talk about gene regulation. So, so there's 2 types of chromatin found in the nucleus of the cell. So you got euchromatin, which is usually loosely bound. The DNA is loosely packed around histones, and it's easily transcribable. Heterochromatin is when it's pretty tightly packed and it's not transcribed. So So euchromatin tends to have high acetylation. Acetylation is when you've added acetyl groups to the histones. You have activation of gene expression. Methylation, you add a methyl group to either DNA itself or histones, and that can reduce transcriptional activity. So let's just go over some, like, the overall kind of overall teams, I guess, gene activation versus gene inhibition. So acetylation can activate genes. Demethylation, so removing methylase or removing methyl groups, can activate a gene. Now, the opposite, deacetylation or methylation, can inhibit a gene's activity. The enzymes that activate are demethylase, which does demethylation, and acetyltransferase, which acetylates. Enzymes that inhibit are methyltransferase, which methylates, and a deacetylase, which removes an acetyl group. Now, you might end up seeing a question that's like, what would a histone deacetylase inhibitor do? Well, you might realize that acetyls are activating, deacetylase inhibits, and a deacetylase inhibitor would activate. So knowing this will help you sort through those triple negatives. Okay, enzyme substrate interactions. So we were talking about this before about DNA polymerase. So you might encounter questions that'll ask you stuff about what amino acids are likely to be in a certain active site. And so you can use that you can use the structure of the substrate to tell you that. So you have a lot of negatively charged groups. You might expect some basic amino acids in the active site. If you have a positively charged group, you might expect a negatively charged amino acid. If you have a bunch of hydrophobic sections of your substrate, you might expect hydrophobic side chains in your active site. Yes, the recording will be available in the post-session email, so just keep an eye out for that. Processes. Okay, so this is going to be it's going to be pretty fast, but you'll be able to go back to this and go back to and look up and study these details. So DNA replication, DNA polymerase, and comparing it to RNA polymerase, and then all the enzymes: primase, helicase, topoisomerase, ligase, leading versus lagging strands, Okazaki fragments. This is all stuff that you should include in your study of this topic. Where does it take place? How does priming work? How are mutations showing up? How are they corrected? What happens to the product? And then how it's different in eukaryotes versus prokaryotes. Transcription, RNA polymerase versus DNA polymerase, transcription factors, promoters versus enhancers, coding versus template strand, sense versus antisense strand. That's super important. They'll ask about that, and knowing which one is which is very important. And then eukaryotes versus prokaryotes. Post-transcriptional modification. Knowing how alternative splicing works is really important. SNRPs in the spliceosome, not as important, but it's useful to know that they're involved in splicing. The 5' cap and the poly A tail, what's their purpose? Where is this taking place? What's the product? What happens to it? And the fact that it only happens in eukaryotes. Translation, ribosomes, structure of ribosomes, the AP and the E sites, eukaryotes versus prokaryotes, codons versus anticodons, and tRNA structure. Membrane transport system is also really helpful to know. The endosomal system, what happens when there's phagocytosis, pinocytosis, macropinocytosis, exo or endocytosis, the ER and the Golgi, what each one does, retrograde transport between the ER and the Golgi, endosomes and lysosomes. Blood flow, the chambers and vessels in order. Smooth and rough ER, good question. Rough ER is where ribosomes can actually dock and where protein folding can occur. So it's called rough because the ribosomes can be docked there. Smooth ER is usually involved in lipid synthesis and detoxification within the cell. And it's called smooth because there's no ribosomes docked on it. Blood flow, knowing the chambers and the vessels in order as far as how they, so not every vessel, by the way, I just wanted to clarify that. You don't have to know all the vessels, just like right atrium, right ventricle, pulmonary arteries, pulmonary veins, left atrium, left ventricle, aorta, and then you come back to the, and then aorta, arteries, arterioles, capillaries, venules, veins. You get the idea. And then where the pressure is highest versus lowest. Glycolysis, you should know, this is important because there's a distinction between glycolysis and the other steps. You should know the enzymes, the intermediates, and their structures, which you can actually learn surprisingly quickly. It's not as hard as it sounds. Inputs and outputs, under what conditions it occurs, oxygen, not oxygen, etc. Regulation mechanisms. So we talked about ATP inhibiting hexokinase. So you should know some of those common regulatory mechanisms for glycolysis. Citric acid cycle, enzymes, intermediates, but no structures. Inputs, outputs, conditions, regulation mechanisms. The electron transport chain, you should know what the relevant proteins are. You should know the electron carriers in order. You should know both their regular names like ubiquinone or cytochrome C and their complex number, so 1, 2, 3, or 4. How many electrons does each one carry? Which ones are proton pumps? Which ones aren't? And where NADH deposits electrons versus FeDH2, why that difference is important, and where they're positioned in the mitochondrial membrane system, because there's a double membrane and the electron carriers are positioned in a certain way. Mitosis and meiosis, what happens in each stage, how it's different between mitosis, meiosis 1 and meiosis 2, knowing the relevant cellular structures, centrioles, kinetochores, chromatids versus chromosomes, and the cell cycle overall. Strategies. So I'm gonna have an overview. I really liked modularizing the information as much as I could. So you can kind of group it up into chunks. So like types of RNA, all the types of RNA. I think I forgot tRNA on that list. Enzymes and DNA replication. That's a module. You have a list of stuff that you can memorize together and associate with each other. So modularizing it can be really helpful because it's a lot of information, but breaking it into chunks can be really helpful. And also making a list of big picture processes that you need to know, and then go into details on that. So, and then you can even group them based on what they do. So cell energy, DNA, cell division, etc. This is a really important one that I go through a lot when I'm tutoring people. The idea of something being true but irrelevant. Every question has exactly one right answer, but usually more than one true answer. So you'll often see answer choices that are, like, strictly speaking, they're true, but they're not related to the question itself. So they're true but irrelevant. So you might have an answer that's like, why, like, so and so is, and then it's dot dot dot, and then it's like property because reason. And every single answer is something because reason. Now, some answers might have a good, like the right characteristic or the right property, but the wrong reason. Some might have the wrong answer, but the right reason. Some might have the wrong answer and the wrong reason, but the one you're looking for is the right answer with the right reason that's relevant to the question. Another thing I want to add that I thought of after I made this presentation was use the answer choices to guide what you're thinking. So for example, if a question is asking about a complicated process, but it's asking only about one aspect of that process, if you look at the answers, you'll realize that all the answers have something to do with that particular aspect of the process. And then when you're looking for an answer to that question, you're not going to look at the entire process and be totally lost. You're going to focus on that exact detail because all the answers have to do with that one detail. So you can zero in on what's important because the answers are telling you what's important and therefore how to answer the question. Okay, I've hit you with a lot of stuff pretty fast. This will all be available as a recording, so don't worry if you didn't catch everything. I just wanted to get through as much stuff as possible so you'd have access to it and I could communicate that to you. So now we have about 15 minutes for questions related to MCAT prep, the exam, strategies, anything we talked about or even didn't talk about in this presentation. Go for it. You can either unmute yourself or put it in the chat. Where can we find the recorded videos on YouTube? So you'll get a link to this video once it's uploaded in the post-session email. Do you have types of keywords that they use in passages? Well, that's a good question, actually, because if you hear that a protein like inhibits the cell cycle or is a key regulatory point on the cell cycle, that kind of hints toward tumor suppressor, although you can never assume stuff. It's a clue. Promote cell growth, promote cell proliferation. That usually is a kind of a hint toward cancer. Does that answer your question? Is UWorld a good resource? I think UWorld is second only to the AAMC, and it even has a couple of advantages to the AAMC. So I would give UWorld 10 out of 10 as far as usefulness for the MCAT. I think we were running a promotion, like a discount through MCAT 101 for UWorld. I don't know if it's still going on. If you're interested in it, you can email us. I'm going to put the email address here in the chat. So yeah, UWorld, fantastic. The best way to use it, I think, is to use it both as a resource for practice and as a resource for collecting topics. So UWorld, what it does is it pretty much covers everything that you could possibly see on the MCAT with its 2,000 questions. And what's really nice is its answers are very detailed, and it also includes like tables or charts related to the topic that the question is about. And so you can just screenshot those tables, put them in your study guide, and then study them. I think it's useful both as a tool for practice and for like figuring out and for even learning content. So it can be useful for both. Okay, let's see. Questions. Multiple topics in a day or in chunks, or one topic a day? Honestly, if you ask 10 people, you're going to get 10 different answers. I would say figure out what works for you. You might try stuff and you're not feeling it, and you might have to do some trial and error. But honestly, it really depends on how you study. I usually would spend a few days on a particular topic and then kind of jump around. And then as I got more comfortable with everything, I would mix it up and do combinations of topics. Effective study schedule for the MCAT. We have some resources on that. If you're interested in that, because that's not my strength. I just kind of winged it, which is maybe not the best strategy for everybody. But I would say send us an email. We'll link you to the guide that we've got. MCAT prep courses. Okay, personally, I am not a huge fan of prep courses because they're very expensive, and as far as bang for your buck, getting tutoring is honestly a better value for your money, because the courses tend to be pretty like one size fits all. I'm not a huge fan of that. And yeah, so I mean, I don't think you need to take a course while you're a sophomore. That's too far in advance. I think personally, I think that the maximum you should prepare for the MCAT is 6 months, no more. Although I would recommend more like 4 to 5. Okay, how would you study for physics? Well, you should attend we'll probably be having a physics session at some point soon, I think. Units are important, but we'll probably be having a session on that. So I will refer you to that upcoming session. Flashcards. Anki, I loved Anki. I use it a lot. I mainly used it for terms, and I would drill with it pretty much every day. I made one for psych. I used that 300-page document from Khan Academy from Reddit, study for psych and soc. I went from a psych novice who only took intro psych to getting a 132. So things can happen if you spend a few months on them with Anki and with that guide. Yeah, every day. So yeah, it took me a few months, but I did go through that entire document. And you can also cut. I usually recommend against using other people's decks because you don't know what they were thinking or focusing on or how they're going to define stuff that might be different from the way you're thinking. I would recommend making your own deck, because then you remember the term from making the deck, and then you remember it again when you actually see the card. So that repeated exposure is going to help you remember. All right. Any other questions? Got 7 minutes-ish. Thanks. I'm glad I could help. Full-length exams. So full-length exams, I usually say don't take more than one every 2 weeks in general, because like otherwise you're just gonna hate yourself. I would say if you get all the AAMC ones. I think that you should only take the third-party full length if you need practice with stamina and getting used to the testing environment. Using them to test your knowledge is kind of a dangerous game. It can be sometimes useful, but they tend to go way in depth, the third-party ones, to scare you into buying more of their shit, like the Princeton Review and Kaplan. So take this with a grain of salt. The AAMC ones, I would say, like if you're taking it in July, start studying maybe this month. Take the sample test first. Sample test should be first. That should be the first thing you do, regardless. That'll tell you where you are. Then I would say maybe take one a month. Maybe take 2 in the last month if you have 2 left. But it's good to spread out your practice tests so you can keep regular tabs on what you're doing. Do your full length exams with your mask on? So it's good practice. It depends on how much your mask bothers you. I personally didn't, because I was just kind of like, screw this. It depends on how much your mask will bother you. I didn't, but if you think it'll help you, by all means, do that. We'll also include the link to the previous session, which was about the overall MCAT, in our post-session email. How many practice questions per day? Well, it depends on what day it is. So I took a reduced course load when I was studying for the exam. And so I don't even remember. Some days were more review, like with Anki. Some days were more practice. I could get through maybe 100 in a day. Maybe some days I do 10, sometimes I do 100. It's honestly like it's kind of how much ever you want to do. You gotta have to balance practice and like content review, which you have to get a feel for it. Would it be all right to take the MCAT later in the year, like September, and apply to the next spring? Oh, absolutely. I took the MCAT last June. I haven't even started applying yet because I'm taking 2 gap years. The MCAT is good for 3 years. So if you want to take it close to your courses, by all means, like do that. There's no requirement that says you have to wait until right before you apply to take it. You're welcome. Jack Weston for cards. Jack Weston is I would say it is a good tool, but sometimes it can be a little janky as far as the difficulty. Khan Academy passages are awesome. I think they might be expiring at some point, and they might be / moving. I don't know the details on that, but Khan Academy passages are great for practice. Some DO schools say MCAT is good for 5 years. Yeah, possibly. I'm not super well versed in how DO admissions works, but that may well be true. Anybody else? You're welcome. One last thing I would say is whenever you start doing a passage or an exam, don't go into it with having already decided that you're not going to understand it. Like I've worked with some students that they definitely know how to do stuff, but because when they start reading the question, they automatically decide that they don't know how to do it and they make some dumb mistake or something they knew, they just get it wrong. So never negotiate against yourself. Look at every practice question as something you either know already or are about to learn. And then you will eventually have way more stuff that you know already and less stuff that you have to learn when you get to the real thing. All right, well, I'm glad this was helpful. Last call for questions before I end the session. Resource review personal statements. I think we do that. Send us an email. Yeah. All right, cool. All right, thanks everyone. Okay, my contact. I'm gonna put my email address right here as well. You can email us at mcat101questions@gmail.com. You can also email me personally if you have any questions. Stay tuned for the post-session email, which is going to have the recording from this session and last session, and stay tuned for future sessions on the other sections.